I'd like to introduce Sophie Nielsen, who gained her BSc Joint Honours degree at Cardiff University in Psychology and Physiology. Sophie graduated from CNELM with the MSc in NTPD. She is currently based in France, where she has set up her own virtual nutritional therapy clinic, specialising in fertility, hormones and brain health. So today, Sophie's presentation is on the link between gut microbiota and Parkinson's disease, a systematic mechanism review with focus on α-synuclein transport. So welcome, Sophie.
So firstly, thank you to Kate and everyone at CNELM for this opportunity to share my research on the link between the gut microbiota and Parkinson's disease. So the population were Parkinson's disease patients. For my research dissertation for the MSc, I included N-acetylcysteine as an intervention, but this was not included in the published paper. Instead, the paper focused on elucidating the mechanisms of pathophysiology of PD, with a specific interest in the role of the gut microbiota and the retrograde transport of pathology from the gut to the brain. So mechanistic markers were included for gut dysbiosis, intestinal permeability, bacterial translocation, so that's microbial products moving from the gut lumen across the intestinal epithelial, inflammation in the gut and the brain, α-synuclein aggregation in the enteric and central nervous system, and oxidative stress. The outcomes were the classical motor symptoms of PD and the non-motor symptoms, namely gastrointestinal dysfunction and loss of dopaminergic neurons in the brain. So this project was a systematic mechanism review to offer a robust systematic analysis of evidence from human, animal, and in vitro studies with critical evaluation of the research hypothesis. So what we wanted to investigate was whether the neurodegenerative cascade begins in the gut. Is gut dysbiosis a mediator? And does pathology spread from the gut to the brain? So this research may help to identify peripheral biomarkers within the enteric nervous system and allow for an earlier diagnosis in PD. And it could lead to the identification of novel interventions aimed at correcting gut dysbiosis, intestinal permeability, for the management or even prevention of PD. So the scope of research. The aims of this project were to investigate whether gut dysbiosis alongside intestinal permeability leads to increased bacterial translocation and overstimulation of the innate immune system. We wanted to investigate the peripheral immune response, so pro-inflammatory cytokines induced by lipopolysaccharides, which can also disrupt the blood-brain barrier, leading to increased α-synuclein uptake, microglia-mediated inflammation, and neurotoxicity. Additionally, whether greater exposure to these bacterial endotoxins may cause excessive α-synuclein expression or even misfolding in the enteric neurons. So our objectives were to provide a summary of pro-inflammatory gut bacteria associated with PD. We wanted to evidence bacterial translocation, enteric inflammation, glial cell activation, and α-synuclein aggregates in the enteric nervous system prior to being observed in the brain, which would suggest a prion-like spread of pathology from the gut to the brain. And research has suggested the dorsal motor nucleus of the vagus nerve as a possible gut-to-brain route. So we wanted to evidence Lewy pathology in the DMV prior to the midbrain region of PD patients, and evidence the activation of microglia in the substantia nigra triggered by the interaction of toll-like receptors with misfolded α-synuclein, which has been shown to promote inflammation and oxidative stress, leading to neurodegeneration. So the neuropathological hallmarks of PD are loss of dopaminergic neurons in the substantia nigra and the appearance of Lewy bodies. Key characteristics of PD are the progressive decline in motor abilities due to the decline of dopamine in the basal ganglia. However, a number of non-motor symptoms are gaining attention due to their more pronounced negative impact on quality of life. These non-motor symptoms are often evident before the classical motor symptoms And gastrointestinal dysfunctions are experienced by most patients, and constipation is known as a frequent premotor symptom. So Brack and colleagues hypothesized that a toxin or pathogen can gain access through the epithelial lining of the GI tract, disrupt the enteric nervous system, and shape alpha-synuclein misfolding and aggregation. And it appears these aggregates can spread in a prion-like manner by the retrograde synaptic transport to interconnected brain regions. Traditionally, neurohormonal and inflammatory factors were seen to primarily mediate communication between the gut and the brain, but more recent research has identified the strong influence of the human gut microbiota on brain physiology and maintaining homeostasis. And it's been termed the microbiota gut-brain axis. Gut microbiota alterations have been found in PD with increased abundance of pro-inflammatory bacteria. The exact nature of these changes are yet to be defined. So this project first looked at human case control studies. As we know, this study design fails to establish causation, so these studies are more appropriately used to generate hypotheses. Most of the case controls adjusted for age and gender, but only half adjusted for BMI and anti-PD medication use. And dietary habits were often not properly controlled for, which could account for the differences observed in the microbiome. Total sample sizes are generally small and from a geographically restricted region, therefore limiting the external validity of their findings. However, all 22 human case control studies demonstrated an altered gut microbiota in PD compared to healthy controls, with results suggesting a pro-inflammatory phenotype is present in PD. And pathobionts are able to produce endotoxins and trigger inflammation in the gut, whereas cellulose-degrading bacteria seem to be decreased in PD, can produce short-chain fatty acids from fermentation of resistant starch and cellulose in the colon. The in vivo animal studies in this review provide strong evidence for the mechanism, but they are restricted by ethical considerations such as the number of experimental animals tested. Nevertheless, high-quality animal studies were found to support the hypothesis of enteric inflammation and denervation in the enteric nervous system, as well as neuroinflammation and microglial activation in the brain. Animal models of PD demonstrate GI dysfunction and intestinal pathology prior to the onset of motor symptoms, as well as an altered gut microbiota composition, which suggests that the direction of pathology is from the gut to the brain. A total of 45 animal and in vitro studies were included, and 8 animal studies, including 2 transgenic models of PD, identified a possible pathway via the vagal nerve for the retrograde transport of alpha-synuclein from the enteric nervous system to the central nervous system. So this review has found evidence to support environmental and genetic determinants of PD, whereby gut dysbiosis alongside intestinal permeability and enteric inflammation may act as a catalyst for alpha-synuclein pathophysiology. So this is the mechanism diagram, which was created after reading review, animal and human literature. So I hope you can see it clearly, but if we start at the bottom of the diagram with gut dysbiosis and the pro-inflammatory taxa identified by human case control studies, alongside intestinal permeability, this can lead to bacterial translocation, so microbial products moving across this intestinal epithelium into the blood, where they can travel to extraintestinal sites. LPS then triggers a peripheral immune response via TLR activation and the release of pro-inflammatory cytokines. These pro-inflammatory cytokines can then disrupt the blood-brain barrier, allowing increased uptake of microbial products and alpha-synuclein into the brain, which in turn triggers microglia activation via the toll-like receptors, oxidative stress, and neuroinflammation, leading to neurodegeneration via various pathways. And finally, if we look at the green arrow, It denotes where antecedents, triggers, or mediators ameliorated pathophysiological mechanisms. And a very pertinent study by Sampson and colleagues using germ-free mice overexpressing α-synuclein demonstrated that the gut microbiota are required for microglia activation, α-synuclein pathology, and motor deficits. I wanted to zoom in on the diagram a bit just to discuss the mechanism relating to α-synuclein transport. So here we have α-synuclein aggregates in the enteric nervous system, leading to autophagy, apoptosis, and denervation. But these protein aggregates can also travel to the brain via the vagus nerve. And this was demonstrated by vagotomy in animal models of PD, which prevented the spread of α-synuclein pathology from the gut to the brain, as well as preventing loss of dopaminergic neurons and behavioral deficits. However, 2 further animal studies using non-human primates found no α-synuclein pathology in the vagus nerve after enteric injection of Lewy body extract. But this difference in findings could be due to the location of the injection site. So one study used the descending colon, which is an area of the gut which does not receive vagal protections. So clinical application. Dopamine modulators are the first-line intervention for PD, but they are often ineffective in the later stages and produce limited benefits of alleviating non-motor symptoms. Nutrition protocols such as N-acetylcysteine, B vitamins, dietary protein, and probiotics are used in clinic to reduce oxidative stress, homocysteine, and support dopamine production, as well as alleviate non-motor symptoms. The etiology of PD is still unknown, which highlights the importance of elucidating the mechanism linking the gut microbiota to neurodegeneration, as it could lead to alternative therapeutic interventions aimed at prevention as well as alleviating symptoms of PD. Intervention research was beyond the scope of this review, but 3 studies investigating probiotics and bacterial metabolites in an animal model or in vitro model of PD were included. 2 of the studies provide evidence for a potential preventative role played by commensals and their metabolites, as they were shown to interfere with α-synuclein misfolding, inflammation, and modulate motor dysfunction. However, one in vitro study found that sodium butyrate increased α-synuclein expression, enhanced autophagy, and activated cell apoptosis in neuroendocrine cells. So this highlights the need for further intervention research into short-chain fatty acids. So future research, we need more large, well-controlled microbiome studies, including control groups taking dopaminergic drugs, COMT inhibitors, and drug-naive PD patients, as well as prospective longitudinal studies in subjects at risk of PD. Future metagenomic studies could provide more information on the functional markers of gut dysbiosis and their impact on the mechanism. And advancing research using machine learning models could prevent novel tools to predict and classify PD based on microbiome data, independent of geographical region. So we would like to see more animal models using oral administration of LPS. This methodology has the potential to investigate the entire mechanism from impaired gut barrier function, bacterial translocation, and the abnormal immune response in the gut to α-synuclein pathology and neurodegeneration. And likewise, animal models using fecal microbiota transplants from PD cases. From our systematic search for the publication, very few studies using an LPS model of PD were found. An LPS model of PD, which includes injection as well as oral administration, could give us more insight into the mechanism, particularly neuroinflammation. And PD is likely a multifactorial disease with a very complex pathological process, with the focus of this review being on dysbiosis and the retrograde transport of α-synuclein. It limited the research possible on inflammation in the gut as part of the mechanism. So a review focusing on peripheral inflammation or immune cell trafficking could clarify the role played by enteric inflammatory markers, glial cell activation, or peripheral immune cell infiltration into the brain on α-synuclein pathology. So that's me. Thank you everyone for your time.